Hello, we're Ed and Rachel from Heart Breakfast.
You've donned your dealy boppers, you've worn your fairy wings and you've done your race for life. You've raised loads of money with all your friends and had a really good time doing it. Now, I suppose you want to know what happens to your money.
Well, it'll come to a place like this. This is the Birmingham Cancer Research UK Centre. Let's go and have a look around. So, that's the lab, that's where it all happens, but we're not allowed in there yet.
Oh no, before they let us anywhere near a pipette or a petri dish, we're going to have to get ourselves geeked up.
Is it called Geeked Up?
I think so.
Okay.
See? Geeked up.
They only had a small.
We're here in the Cancer Research UK laboratory with Dr. John O'Neill and his glamorous assistant.
And we're going to have a go. So, Dr. John, what are you working on now?
Well, what I've got here are cancer cells growing in a flask, and the ability to grow cancer cells in a flask is really important to us because we can use these for our research instead of people.
Can I have a look?
Yeah, sure.
Ooh. Wow! They look a little bit like your wallpaper, Ed.
Is this lunch then, Becky?
Well, actually, we're going to get you to extract some DNA from the strawberries.
Brilliant, how do I do that?
You put it in the bag and you mush it all up.
That doesn't sound very scientific.
No big bits in there.
This looks like a fun bit. Is this the best bit of your job, mushing strawberries?
Well, we don't usually use strawberries. We normally use cells and we take DNA from the cells. But we're going to get DNA from strawberries today, so keep going, keep mushing.
You ever wonder what scientists did all day?
And then we have the big bits at the bottom that have been spun to the bottom, and our liquid with our DNA in at the top. 30.
Right, ethanol in here.
Yeah.
How many squirts?
And if you do it up to the 30, you actually have to squeeze the bottle.
Okay.
30 mils on the thing.
So everything at the top is DNA?
All the white stuff is, yes.
Wow, so that is strawberry DNA. That's amazing.
So Ed's extracted the DNA. Why is it so important in your research?
Well, we normally extract DNA from cancer cells and normal cells to compare them. And you get mutations in DNA that cause cancer. So we tend to have a look to see what differences there are in these cells. So what we're going to do now is actually load some DNA into a gel, and it will separate out and show us the differences between the cells that we've put in.
Can I compare some DNA?
Yes, if you take the pipette and you put a tip on it, and now press down on the plunger and get some liquid. This is how we measure out small quantities of liquid. And if you go over to the gel and put it in one of the wells,
That's whereabouts? That right?
there, yep, and head down and press down on the plunger. Perfect. And if you discard what's left into the pot using the other button, and then we have all our samples next to each other, and we can compare differences side by side to see how active the DNA is and to see whether there's been a mutation.
So once you've spotted the mutations in the DNA, what does that mean? What do you do?
Well, single mutations aren't necessarily cancer. Cancer's when you get multiple mutations on top of one another, but a single mutation allows the cells to get out of control. And you can have these precancerous tumours that aren't necessarily malignant, they can't spread. But if we can identify these cells at the stage when they've only got one or two mutations, we can actually take them out before you actually get cancer. So that's what we're attempting to find, single mutations that lead up to the onset of cancer.
So that's it, you've now seen exactly where your money goes.
But sadly, 1 in 3 fundraisers don't send their money in.
1 in 3. So unless you want this whole project to be left in the hands of me and Ed, please do send your sponsorship in.
Thanks, girls, for all the hard work you put into raising money for Cancer Research UK.
And thanks to all the other fundraisers. Without your cash, none of this would be possible, so big thanks.
Thank you!